All right, guys, this might not be the best audio quality because I'm at school, but here we go with our last section of notes in Unit 2. So this covers topics 2.10 and 2.11. And the notes that we're going to encounter today are mostly related to what we saw when we did our video study Thursday and Friday last week in class. So let's start with sort of the formation of the first cell. So keep in mind, we learned last week that early Earth's environment was very, very different than it is today. It was very inhospitable to life, very violent. And I've noted a couple of specific things that were very different. So early Earth's environment most likely had some pretty poisonous gases that made up our atmosphere. And those gases were coming from volcanic eruptions. The sun's UV radiation was so intense that it would kill any living thing, which gives rise to that understanding, present day understanding, that most likely life was created deep, deep, deep in the ocean where there was no sunlight available to kill that first living cell, and around these hydrothermal vents where you could find some chemicals to use as energy. Most of Earth was most likely covered in water. And again, very violent volcanic eruptions and lightning storms most likely existed. So the reason that we go over this is because of the famous experiment that we talked about. Stanley Miller and Harold Urey in the early 1950s decided they wanted to see if they could test another scientist's theory by building a closed system in a lab that could mimic early Earth's environment. And so knowing what early Earth looked like is super important to understanding their experiment and why they chose the aspects and the components to put in the model comes from the understanding of early Earth's environment. Now, keep in mind, they were testing Oparin's idea. So the video that we saw on Thursday or Friday introduced Oparin and how he thought, this is a very simplified version right here, how he thought that if we could just create the beginning of life, if we could create some carbon-based molecules, eventually macromolecules, those would spontaneously form into cells. And so what Miller and Urey were trying to do was test whether they could make kind of the base of Oparin's idea, if they could make some of those monomers and eventually some polymers that could self-form into cells. So we're continuing in the same section of notes here, kind of scrunch your writing. We've got one more slide to discuss this model. So the experiment that they ran, they put together this contraption, and we saw video over this, so I know I'm going quickly, but this shouldn't be terribly new to us as an understanding. So in the experiment, or in the model, we have to look at the different components and what they were trying to replicate. So the water right here in this chamber, the water was here to model oceans and the fact that early Earth was most likely primarily covered in ocean. Then they heated the oceans to induce the water cycle. So anytime you heat up liquid water, it will vaporize and rise and collect. And so here is the next chamber where those water vapors collected. And this was full of some very interesting gases. These gases are the gases at the time they thought existed in early Earth's atmosphere. So this chamber right here represents the atmosphere. And they introduced an electrical spark into this chamber that replicated the lightning storm. They needed a source of energy, and they knew that lightning storms, or at least they theorized that lightning storms existed. So they introduced electricity to mimic the lightning storms. Then they had to cool all of this down. So this chamber gets pretty hot when you are zapping electricity into it. And so if you want these components to kind of trickle back down to, quote, Earth, our oceans, then you got to cool it down. And so they wrapped a condenser, which just circulated cold water over this section of tubing. And that let all of these gases start to cool down and fall down in the form of water. So this mimicked rain coming back to the ocean or the primary water source. And then, of course, they collected a sample after they let the system run for about a week, our video said. The water turned an interesting color, and so they sampled out of that water. And what they found were amino acids. So as a reminder, amino acids will link together to form the polymer that is a protein. So in their experiment, they were able to create amino acids based on simple components of early Earth's environment. Now, there is a pretty significant critique to their experiment, and that is they may not have gotten these gases exactly correct. And so that changes some of the chemical compositions that would have been available to create whatever ends up being created. And so many experimenters, scientists have come along in their footsteps and have redone this experiment using new information. And so recent experiments actually reveal that most likely the first monomer that was created in early Earth's environment was not an amino acid, but rather a nucleotide. And therefore, the first polymer that was formed had to be made of nucleotides. Now, present day theories suggest that RNA was that first molecule because it was more simplistic compared to DNA. Remember, RNA is single-stranded right here. And because we know there are many different kinds of RNA present day, the idea that RNA is a pretty versatile molecule that can do a bunch of stuff also gives a little bit of credit to the idea that it was most likely first and not DNA. And so this is called the RNA world hypothesis, the idea that RNA came first, because it could do lots. It's sort of like a Swiss Army knife. It can do lots of different things. It could self-replicate. It could store a code, which eventually could become a genetic code that holds, the code for the creation of proteins and a next macromolecule. But also we know that RNA can induce metabolism. And so the idea that this first cell needed energy of some sort, it needed to be able to metabolize that energy, and RNA can do that. Now, this is stepping a little bit into something that is new. So the idea here is that most likely RNA formed first, and that RNA was used to create some proteins, and then most likely lipids came next. And because this was all happening in a watery environment, we know that lipids form a bit of a sphere when dropped into water because they are amphipathic. And so we get this very primitive, early version of a cell. And that continues to evolve until we get a prokaryotic cell, which we have talked about. If that word rings any bells, that would be a cell that over time would be something we would call today a bacterial cell. And so once the first cell is formed, that cell needs to be able to use energy. So this next section of your notes is new information, and we will continue to go over that this next section when we meet today or when we meet this week, whether that's Monday or Tuesday. But we're talking about metabolism, the ability to use energy to grow and reproduce, characteristics of a life, of cellular life. And so the reality is we cannot talk about metabolism without considering early Earth's environment was weird. And so most likely that first metabolic strategy supported what the environment looked like. And so once the first cell, which we think was a very simplistic single-celled prokaryote, formed in water, we see metabolism strategies emerge. So most likely chemosynthesis was that first metabolism strategy. We talked about this last week. Chemosynthesizers are organisms that can use chemicals to create their own energy source. They are autotrophs, but they're not using the sunlight, they're using chemicals. And so this gives further support towards the idea that we believe life formed in these deep sea hydrothermal vents that were spewing out all of these really intense chemicals. The first prokaryotic cells were able to utilize those chemicals and survive and grow and reproduce as cells, as living organisms. Now, anytime we talk about metabolism, if an organism is consuming or creating energy to be consumed, even if it's by themselves, they're going to have some kind of a waste product. And so the idea here is that chemosynthesizers ruled Earth, but as they metabolized in this unique chemosynthetic way, they produced things, byproducts that actually changed their environment, changed the water source, changed the atmosphere even, and paved the way for our next metabolism strategy. So up here in our picture, this is that reminder that chemosynthesis was probably the first primary mechanism for these simple single cells to be able to harvest energy. In this case, to be able to make their own energy to kind of support their own life. But their byproducts changed their environment. And most notably, we think that it gave way to photosynthesizers. So the chemosynthetic organisms' products most likely helped clear up the atmosphere and make it safe for organisms to now use the sun. So the UV light is no longer killing living things. And so photosynthesis becomes the new metabolic strategy. Photosynthesizers are way more efficient at creating their own energy than chemosynthesizers are, which was the benefit here that supports the idea that now the majority of life becomes photosynthetic. It becomes an advantage if you can photosynthesize because you have an abundant source of energy and you're more efficient at creating your own energy. And so if you know anything about the process of photosynthesis, you know that you are taking in carbon dioxide, and one of the products that you are releasing is oxygen. So this is a very interesting shift in our atmosphere, which had no oxygen in the form of O2. So now, because photosynthesizers rule the earth and are primarily using photosynthesis as an energy source, we see again another shift in the components of the environment, which paves the way for our last primary metabolism strategy here, which is called aerobic respiration. And so we actually are aerobic organisms. We take in oxygen and we use that oxygen plus the food that we eat. We are consumers, so we're seeing a shift here. The bacteria are no longer creating their own energy, but are rather consuming other bacteria for energy. And so taking in oxygen, because it's rich in the atmosphere and consuming their own energy, they're able to make ATP. And so this is by far the most efficient metabolism strategy, which is why it is the primary metabolic strategy of most eukaryotic organisms like us present day. So prokaryotes really mastered it. And all of this, I'll say, all of this metabolic evolution was occurring as prokaryotes, single-celled organisms, dominated the earth. So we have yet to even see the existence of a complex cell, a eukaryotic cell. But as single-celled prokaryotes, really, really simple bacterial cells, we think that this is the evolution of their metabolism strategy, which again is the story of something called coevolution. As the cell changes and produces, the atmosphere will change. And so the cells then, to fit into that new atmosphere, are forced to evolve again. And their evolution, new strategy for metabolism emerges, new products are released into the atmosphere, and again we see the atmosphere shift. Our pictures over here, this is our chemosynthetic prokaryote that gives rise to our photosynthetic prokaryote. This is a really common photosynthetic prokaryote that still exists in high counts today. It's called cyanobacteria. And then eventually to our aerobic prokaryotes. Now, this whole thing that I just talked about is something called the oxygen revolution, where we see a pretty intense shift in early Earth's environment. Used to be full of lots of crazy chemicals. Now it's full of a lot of oxygen. So this video will help us very quickly to kind of recap that idea. Watch along with me.
There's an organism that changed the world. It caused both the first mass extinction in Earth's history and also paved the way for complex life. How? By sending the first free oxygen molecule into our atmosphere. And they did all this as single-celled life forms. They're cyanobacteria, and the story of these simple organisms that don't even have nuclei or any other organelles is a pivotal chapter in the story of life on Earth. Earth's atmosphere wasn't always the oxygen-rich mixture we breathe today. 3.5 billion years ago, the atmosphere was mostly nitrogen, carbon dioxide, and methane. Almost all oxygen was locked up in molecules like water, not floating around in the air. The oceans were populated by anaerobic microbes. Those are simple unicellular life forms that thrive without oxygen and get energy by scavenging what molecules they find. But somewhere between 2.5 and 3.5 billion years ago, one of these microbial species, probably floating on the surface of the ocean, evolved a new ability, photosynthesis. Structures in their cell membrane could harness the energy from sunlight to turn carbon dioxide and water into oxygen gas and sugars, which they could use for energy. Those organisms were the ancestors of what we now call cyanobacteria. Their bluish color comes from the blue-green pigments that capture the sunlight they need. Photosynthesis gave those ancient bacteria a huge advantage over other species. They could now produce their own energy from an almost endless supply of raw ingredients. So their populations exploded, and they started polluting the atmosphere with a new waste product, oxygen. At first, the trickle of extra oxygen was soaked up by chemical reactions, iron, or decomposing cells. But after a few 100 million years, the cyanobacteria were producing oxygen faster than it could be absorbed, and the gas started building up in the atmosphere. That was a big problem for the rest of Earth's inhabitants. Oxygen-rich air was actually toxic to them. The result, about 2.5 billion years ago, was a mass extinction of virtually all life on Earth, which barely spared the cyanobacteria. Geologists call this the Great Oxygenation Event, or even the oxygen catastrophe. That wasn't the only problem. Methane had been acting as a potent greenhouse gas that kept the Earth warm. But now, the extra oxygen reacted with methane to form carbon dioxide and water, which don't trap as much heat. The thinner atmospheric blanket caused Earth's first, and possibly longest, ice age, the Huronian glaciation. The planet was basically one giant snowball for several 100 million years. Eventually, life adjusted. Aerobic organisms, which can use oxygen for energy, started sopping up some of the excess gas in the atmosphere. The oxygen concentration rose and fell until eventually it reached the approximate 21% we have today. And being able to use the chemical energy in oxygen gave organisms the boost they needed to diversify and evolve more complex forms. Cyanobacteria had a part to play in that story too. Hundreds of millions of years ago, some other prehistoric microbes swallowed a cyanobacterium whole in a process called endosymbiosis. In doing so, that microbe acquired its own internal photosynthesis factory. This was the ancestor of plant cells, and cyanobacteria became chloroplasts, the organelles that carry out photosynthesis today. Cyanobacteria are still around in almost every environment on Earth: ocean, freshwater, soil, Antarctic rocks, sloth fur. They still pump oxygen into the atmosphere, and they also pull nitrogen out to fertilize the plants they help to create. We wouldn't recognize life on Earth without them, but also thanks to them, we almost didn't have life on Earth at all.
Okay, and we will continue when we meet this week discussing that whole idea of one cell engulfing another cell. And that's really what eventually gives us a complex eukaryotic cell like the video showed a plant cell. So all of that is to come this week. You are done with this section of notes, and I will see you in class.